Everybody, welcome to ClubEvMed. My name is Charlie Nunn. I'm at Duke University in evolutionary anthropology and the Duke Global Health Institute, and I run the Triangle Center for Evolutionary Medicine. And it's great to see so many people coming in to today's ClubEvMed. A lot of you have been to ClubEvMed before, but let me just remind you that it's organized by the International Society for Evolution Medicine and Public Health. And I want to encourage you to come to our annual conference that's July 8th to 10th in Nashville. It looks like they're still accepting abstracts. At least that's up on the website as such. So if you'd like to submit an abstract, please do. And also, please consider submitting articles to the Society's journal, Evolution, Medicine, and Public Health. ClubEvMed's also organized by a bunch of other centers, which I'm going to paste in here.
Good
I'm not going
afternoon.
to name them all, but we have a lot of other centers that are involved in organizing ClubEvMed. So thank you all for your partnership on that. And one last thing, these centers are also involved in organizing a workshop this summer, which again, I'm pasting in. It's called the Evolutionary Medicine Summer Institute, or MC. And this summer, we're organizing the workshop around a topic, and that topic is overcoming resistance, harnessing evolution to control cancer, infectious diseases, and pests. It's going to be held May 19th to 23rd in Raleigh at North Carolina State University. And if you're interested in coming and joining us, please click on this link. It'll take you to where you can find more information, including the application portal. And we'd love to have a good showing of people there. And so please do apply if you're interested in joining us. We've got a great lineup of instructors. You can learn more about that on the website. Okay, with that, I want to introduce our speakers today. We have two people speaking. We have Jesse Shapiro. He's an associate professor of microbiology and immunology at McGill, where he and his team use genomics to understand the ecology and evolution of microbes, including pathogens, their evolution within patients, and interactions between those pathogens and the resident microbiota. McGill is also where Jesse got his undergraduate degree before going to Oxford, and then on to MIT and Harvard. At MIT and Harvard, he worked with Eric Alm and his PhD, and then Pardis Sabeti as a postdoc. I got to know Jesse when we overlapped at Harvard. He gave a couple guest lectures in my evolutionary medicine class, and we've been in touch since then. I was really excited to see this paper come out, which he's going to talk about. And I'll paste a link in to the chat just so everyone has it. He's just done great things since his time at Harvard and MIT. That includes holding a Canada Research Chair. The other speaker we have today is Abu Sayeed. He's a research scientist at the University of Florida. At the time, the paper, let me share that paper with you guys so you have it. At the time this paper in Science was published, he was in Eric Nelson's lab, and there's the link to it. He received his PhD at University of Florida in public health. He's a co-author on this paper. He was working at Eric Nelson's lab, and now he's working on cancer genetics. So welcome, Abu Sayeed. Really great to have you here, and Jesse. And with that, I'll turn things over to you guys to give your presentation. I forgot to ask Jesse, would you like questions as you go, or would you rather have them at the end?
Either
Or
is fine. So I don't know if there's a tradition in this group for interruptions or less interruptions. I'm fine either way. So
okay.
Yeah.
If we see questions in the chat that look like they would be, good to have conversation about at the moment, like a clarifying question or something, we'll interrupt you and go from there. Okay.
Sounds great. Yeah, well, thanks so much, Charlie, for the invitation. And yeah, as Charlie said, we've known each other for a little while, and I really appreciate your support of our work. And yeah, it's great to see you virtually. I was supposed to be at Duke for a seminar next week in person. It will unfortunately be over Zoom due to NIH.
Oh, okay.
Funding issues. So anyway, I'll be at Duke for a Zoom again.
Well, I knew about that. I didn't know it was going to be a Zoom though. So
yeah, it was planned in person, but yes, this was a funding situation. Anyway, but here, I'll just share my screen. Do you see?
Okay, we're seeing the
presenter
preview presenter mode. Yeah.
Okay, let's redo this. And you
And I'll
know what?
just say we should have put a link and the citation on the advertisement. We'll make sure that goes up on the YouTube version of this
Okay.
talk, just so everybody has that easily.
All right. Are you just seeing my
That's perfect.
regular slides there? Okay, great.
Great.
Okay, so this will be a talk in two parts. So actually, feel free to interrupt with quick questions, but I'll sort of end and there we can take questions there, and then Sayeed will actually has a separate short slide deck. And I'll be talking more about the coevolution within patients, and Sayeed will be talking about the improved diagnostics part. So a little bit of two in one. And we're going to be talking about cholera. So for those of you who don't know, we are in an ongoing cholera pandemic. These are the affected countries. More red is more cases. These are cases per 100,000 population. See a lot of hotspots in Africa. In Yemen is a huge ongoing outbreak. Small little dot over on the other side of the Atlantic in Haiti. There was a big outbreak following the earthquake in 2010, which is thankfully resolving, but there's some blips along the way. The endemic region that we'll be focusing on today is around the Ganges Delta and Bangladesh, and this is an area where cholera is essentially always simmering and, if not erupting, into large outbreaks. So this is a diarrheal disease, severe dehydration and risk of death, high mortality rates if not treated. And it's a bit of a tragedy where we know how to treat this with rehydration and antibiotics for severe cases. But really with sanitation, it's preventable as well, right? So this is really a failure of sanitation, of public health. It happens during war and conflicts, earthquakes, whenever all of these systems that in high income countries we can take for granted, very quickly we can be susceptible to cholera. This is just looking at the number of cases. This is all data from 2024 from WHO. So you can see that these cases are dominated by the Eastern Mediterranean region. So going back to the map, this is actually referring to, I think, Yemen and outbreaks actually in the Middle East. And in light green at the bottom is Southeast Asia. And I just want to highlight that these are probably underreported in many cases. And as we get into diagnostics and actually the importance of properly counting cases so that we can respond with public health is incredibly important. So these are undercounts, but we are talking about hundreds of thousands of cases per year, right? So this is showing per week with a lot of morbidity and mortality that accompanies this. Second thing I want to introduce are phages. So for non-microbiologists, quick introduction, these are viruses that infect bacteria. And phages are probably the most diverse and abundant entities, organisms on Earth, because they outnumber bacteria by sometimes orders of magnitude. And phages can be quite specific. So we're going to be talking about cholera phages, and there are different cholera phages. Cholera, so Vibrio cholera, this is the bacterium that causes cholera, has diverse interactions with phages. So through the lytic cycle, and I'll be in this talk, we'll be talking mostly about lytic or virulent phages that basically blow up bacteria. This is a strong selective pressure. So bacteria are under selection to either resist these phages or die, be killed, right? So they're acting like a classic predator. In the lysogenic cycle, these viruses can actually integrate themselves into the bacterial genome and are thus a vector of horizontal gene transfer. And in the case of cholera, this is this can have dramatic effects where actually the cholera toxin, which is what makes Vibrio cholera into a pandemic and very virulent pathogen rather than a relatively benign diarrheal disease, maybe opportunistic human pathogen that mostly lives in the environment, is actually a lysogenic phage that integrated into the bacterial genome, bringing with it the cholera toxin, which is really the key virulence factor. So I'm not going to talk about that integrated phage. I'm going to talk about lytic phages, but just to let you know that there are really these diverse interactions between bacteria and phage, and Vibrio cholera is really a great example of that. So we have known about phages for over 100 years. So this is a paper by Darrell and Malone doing some work in India, and this was published in 1927. They refer to this as the cholera bacteriophage inquiry. They're working in India at the time. And so we've known about these phages for about 100 years, and we've also known about phage therapy, so that phages can actually be useful in treating disease. And this is just a table from this paper. And what you can see in the first column here, and this is sort of a small clinical trial where they're actually treating cholera cases with bacteriophage. And sorry, in the first column are cases not treated by phage, and you can see a very high mortality rate, 13 deaths out of 16 cases, and really, really good success rate treating people with phage, where out of 12 cases, that went down to just 1 death. So small numbers, also, definitely not double blind, definitely not meeting our current standard of a clinical trial, but really showing some very interesting results about the potential of phages to treat disease about 100 years ago. So what's changed over the past 100 years? Well, a few things. We're in the era of genomics, so we can look at phage bacteria interactions in really intricate detail. That wasn't possible 100 years ago. And we've also entered the era of antibiotics, where phage are not the preferred treatment for bacterial infections. We are in the antibiotic era. So starting in the 1940s, discovery of successive antibiotics has been transformative in medicine, as I think this audience knows, and as this audience will also certainly know, has selected for successively increasing antibiotic resistance. And this is true in Vibrio cholera, just as in pretty much every other bacterial pathogen that we know. So today we're going to discuss the interaction between these three partners, three entities: Vibrio cholera bacteria, lytic phage, and antibiotics. So for the purposes of this talk, I'm going to consider antibiotics as a selective pressure on bacteria, so just a molecule that kill bacteria and select for resistance. I'm not going to consider antibiotics as an evolving entity, although recognizing that antibiotics are produced by microbes, and so over million-year timescales, we can actually consider antibiotic production as an evolving trait. I'm going to put that aside and just consider these molecules as a static entity that clinicians will prescribe. Phages, of course, as I've already introduced, are a selective pressure. I'm going to focus on lytic phages that just infect and kill bacteria, not the ones that integrate into the genome. And bacteria and phages can enter into this co-evolutionary cycle where both impose selective pressures one on the other in this loop. And the main question I'm going to be asking is, how do these interactions play out in real patients, right? So this is a nice cartoon, but really, do we see evidence, and how does this really work in the real world? So first, do phages impact disease severity? Do we actually see evidence that people with more phage actually have fewer cholera symptoms because phages are suppressing bacteria? Is there evidence for coevolution in patients, so on relatively short timescales? And the third part that Sayeed is going to address is, can phages actually improve rather than tarnish the performance of rapid diagnostic tests, which are really needed to properly count cholera cases so that we can respond appropriately through public health measures. So this is the team that undertook this work, not showing Sayeed and myself, and not showing the whole author list of this study, but really the main players. And this is going to serve as my acknowledgement slide. So I'll take a minute to acknowledge Naima Mahdi, who is a PhD student in my group, who is continuing on a postdoc. She's the lead author. This is work in close collaboration with Dr. Fayouzi Qadri, pictured here, and Dr. Ashraf Khan at the International Center for Diarrheal Disease Research in Bangladesh, and Dr. Eric Nelson, Sayeed's PhD advisor and a microbiologist and clinician scientist at the University of Florida. Please alert me if there is a question in the chat or anything like that.
We're good so far.
Okay,
But
all right.
please do post questions and comments in the chat.
Okay, yeah, sounds good. It's yeah, and just holler because I don't see it easily on my screen here. All right. So what did we do here? So doctors Qadri and Khan undertook a surveillance of diarrheal patients over 1 year across Bangladesh. They collected just over 2,500 stool samples. So this was for people who came into the hospital with diarrhea. They also did, and this was in Eric's lab, liquid chromatography mass spectrometry to measure antibiotics in stool. This is an important step because in Bangladesh, like in many parts of the world, people can self-medicate by going to the corner store, what here in Montreal we call a dépanneur or a bodega, or basically for a few cents buy some pills to self-medicate. So what was prescribed at the hospital isn't necessarily what they were exposed to, and this is not even taking into account possible exposures in contaminated water, various unknown exposures. So we're trying to measure quantitatively what patients are actually exposed to rather than what they self-reported or what was prescribed. And this is quite important in this particular setting. What we did in my lab was metagenomic sequencing. So basically shotgun, unbiased sequencing of DNA extracted from stool. And we did this for 344 samples that had some evidence for a Vibrio cholera infection. So either PCR or culture for Vibrio cholera, or one of 3 lytic cholera phages that are named ICP1, 2, and 3. I'm going to focus on ICP2, which is just the most prevalent of these phages. And so the metagenomic sequencing gives us a relative quantification of Vibrio cholera and the phages and other bacteria in the microbiome that I'm not really going to talk about very much today, as well as the potential to look for single nucleotide variants, point mutations, and variations within the VC genome or the ICP1 genome, which I'll get into in a bit. And I'll abbreviate Vibrio cholera as VC throughout. So we can estimate the relative abundance of these partners. We also did this with qPCR, quantitative PCR, and this correlates well with the metagenomics. So I'm just going to refer to the metagenomic data. All right, so first question, is the ratio of phage to bacteria associated with disease severity? So I'm going to picture this as a log ratio on the y-axis here, with higher values basically having the phage take the upper hand, and lower values is Vibrio cholera, the pathogen, predominating. And our expectation here is that more phage suppresses the pathogen and might be associated with better disease outcomes. And indeed, this is what we see. So patients with milder disease severity, so this is measured as milder dehydration, do have a higher median phage to bacteria ratio. So these differences are statistically significant. You can see that there's quite a lot of variation, right? So this certainly is not explaining everything. There's other host factors, microbiome factors, but there is an effect here. And just drawing a threshold of 1 to 5 phage to Vibrio cholera, we can make a very simple classifier of non-mild dehydration, so separating mild from non-mild, with a decent sensitivity of 85%, but a pretty poor specificity of 53%. So, we wouldn't actually want to use this as our sole diagnostic of disease severity. There's other information we can use, and this wasn't really the goal of this study, but going forward, we are interested in, what's the minimal amount of information that we could use to improve our diagnostics for not just does someone have cholera, but are they likely to have mild or more severe disease. So this just kind of shows that potential. So next we wanted to look at this interaction between antibiotics and phage. And to do this, we considered a bunch of statistical models where our response variable is the relative abundance of Vibrio cholera. And we're predicting this based on the measured concentration of antibiotics in stool, the relative abundance of phage, ICP1, and potential interactions between antibiotics and phage. And we found a most parsimonious model that includes one antibiotic of 5 that we considered. It turns out that azithromycin is the one that has a significant effect. And also the interaction between azithromycin and phage, ICP1, is significant in this model. So this is what the model effects yield. So this is, first of all, just the effect of azithromycin. Basically, azithromycin is an effective antibiotic against Vibrio cholera. So we see Vibrio cholera abundance declining at higher exposures to azithromycin, right? So this is showing this drug is basically working, which is kind of nice to see, and interesting to see that this does seem to be the most effective drug compared to others, ciprofloxacin and doxycycline, that were also detected but don't have a significant effect. And this is the interaction between phage and Vibrio cholera. So we still have Vibrio cholera on the vertical axis, and then on the horizontal axis, we have the phage, ICP1. And the color code is azithromycin. So as we go from 0 azithromycin in the black curve down to a higher exposure to azithromycin in yellow, we have this sort of quadratic function, right? This kind of peak it goes up and down, that then gets flattened at higher doses of antibiotics. And the way we interpret this is this quadratic is sort of indicative of a predator-prey dynamic, right? Where we have phages tracking positively with bacteria. So, they're an obligate parasite of the bacteria, so they will be positively correlated. But at one point, they'll decouple, where the phages will overshoot and the bacterial populations will start to crash. So we get this negative correlation as phage populations basically overshoot their prey. So this is how we interpret this quadratic. But the quadratic basically gets squashed as you ramp up antibiotics. Antibiotics are almost like a really blunt hammer that just suppresses the bacteria and kind of trumps this more complex relationship between phages and bacteria. So this was interesting to see because you could have imagined different types of interaction or perhaps no interaction. This is a relatively simple interaction that we see between phages and antibiotics. So next, we wanted to get into the more evolutionary angle. So we know that there are resistance genes that bacteria encode to protect themselves against phages. And one of them that's been relatively well studied, or at least since 2021, has been recognized to be important in protecting Vibrio cholera against phages like ICP1, is this integrative conjugative element. So this is 100 kilobase plasmid that integrates into the Vibrio cholera genome, and it encodes a bunch of different genes, and it comes in different flavors. So these, the relevant ones here are IN5 and IN6 that have different hotspot regions. So these differ in their gene content for various different anti-phage systems. So you don't have to, won't get into the details of these, but Vibrio in the IN5 variant encodes a Breck system. the N6 encodes some different restriction modification systems. So these are just different anti-phage systems. So we know about these elements, but we didn't really know, are these relevant within patients, right? Are these actually protective? Do they vary? So we asked the question, are these ICE variants associated with phage suppression within patients? And again, we're going to look at the phage to bacteria ratio in patients that have different ICEs that we're able to detect with our metagenomic sequence data by mapping our sequence reads to these different ICE variants. And what you can see here is that there is an effect that goes pretty much in the predicted direction. And in fact, ICE and ICP1 interact to predict dehydration status. So the first thing to notice is that in the absence of an ICE, right? So when Vibrio cholera does not encode any resistance genes, we see phage kind of tending to get the upper hand and being more abundant. So the ratio of phage to bacteria goes up. So that is what we would expect. And it seems like N5 is the more effective variant, or is most associated with a lower phage to bacteria ratio, where bacteria stay quite abundant, and it seems like Vibrio cholera are well protected, with N6 being kind of intermediate. And if we run some stats on these, so a binomial mixed model, there is a significant interaction between the ICE type and ICP1 phage. And the nature of this interaction is a relatively intuitive decision tree, where at high values of the ICP1 to Vibrio cholera ratio, this predicts mild dehydration. So I showed you this before. So more phage suppresses the pathogen. We have milder disease. Unless Vibrio cholera encodes an ICE defense element, in which case the patient is more likely to be not mild, right? So this is intuitive. I know these are this is like a nested decision tree, but this all makes sense with what we know about biology. Now, there's still quite a lot of variation here, right? These points are quite scattered. So ICE protection against ICP1 must be imperfect. And we hypothesize that there are probably some other resistance mechanisms at play. So we just decided to use our metagenomic data to look for point mutations in the Vibrio cholera genome as potential other resistance mechanisms. So here we're asking the question, does the phage ICP1 select for single nucleotide variants, SNVs, in the Vibrio cholera genome? So again, we're using our metagenomic data, mapping the short reads to the Vibrio cholera genome and looking for point mutations. And here, again, what we're looking at, this is a model fit with our response being the mean SNV frequency in the Vibrio cholera genome as a function of the interaction between phage ICP1, the resistance factor ICE, and the mutation type, whether it's non-synonymous, synonymous, or intergenic. And so this is the model fit for non-synonymous SNVs. And what you can see is that we see this effect where in the absence of an ICE, so when Vibrio cholera does not encode this major defense element, we see that ICP1, as ICP1 goes up along the x-axis, we see higher frequency non-synonymous point mutations in the Vibrio cholera genome. This is not true in the black and gray lines here when Vibrio cholera does encode an N5 or an N6 ICE anti-phage resistance element. So it seems that when Vibrio cholera doesn't already have a resistance factor in its genome, it's under selection to find something else, essentially, that these various non-synonymous mutations are increasing in frequency in response to phage. This is not the case for synonymous mutations. So again, this supports the idea that this is the action of natural selection that's specific to coding variants, so non-synonymous mutations that change amino acids, and we don't really see an effect for synonymous mutations. We also don't see a significant effect of antibiotics on the number or frequency of SNVs. So this is a negative result, which is kind of interesting and goes with the idea that antibiotics are maybe a blunt hammer that really just suppress Vibrio cholera without giving it the chance to evolve, but that phages are maybe an intermediate enough selective pressure, or a selective pressure that tracks slowly with the growing Vibrio cholera population and actually gives it a chance to adapt. Next little thing to note is that most SNVs seem to occur in mutator populations of Vibrio cholera. So when we have these non-synonymous mutations increasing in frequency, this tends to be in populations that have an error, that have some kind of defect, so a non-synonymous change in DNA repair genes, and where mutation rates are going up from 1 or 2 SNVs per patient to 10 or 100, so order of magnitude increases. Which is kind of interesting. People have noted mutator populations in various different bacteria, but this is consistent with phage maybe being an important selective pressure where these mutators are occurring. So just closing this co-evolutionary loop and asking, does Vibrio cholera select for point mutations in the ICP1 genome, right? So we saw the bacterial side of evolution. Here is the phage size, the phage side. And just a caveat that we could only do this in about 45 samples where we had enough metagenomic reads mapping to the phage genome to do this. So we don't have a lot of data points, but we do see an interesting effect where as the phage bacteria ratio goes up, we see an increase in mean non-synonymous SNV frequency in the ICP1 genome. So basically, as phages are getting more successful, as they start to dominate the bacteria more, there are non-synonymous SNVs that are increasing along with that. And this is the case in IN5 samples only. So recall IN5 is the most effective ICE at suppressing phage. So this seems to be when phages are being suppressed generally effectively, they're responding with an increased non-synonymous point mutation frequency. And we don't see these correlations in the absence of an ICE or an IN6, which seems to be less efficient at suppressing phage. And this is not true for synonymous or intergenic mutations, also pointing to selection on the phage. All right, so just to wrap up with a bit of a cartoon here for this part, we see in patients with severe dehydration a dominance of the pathogen Vibrio cholera over ICP1. This is associated with ICEs, so this resistance element in the Vibrio cholera genome that seems to be effective at suppressing phage. And when phages are under this pressure from ICE, they respond with an increased frequency of non-synonymous point mutations. On the other hand of the story, in patients with milder dehydration, we have a dominance of the phage, ICP1. So phage are exerting a strong selection pressure on Vibrio cholera, and that tend not to have an ICE resistance factor, and they are under selection to perhaps become mutators and accumulate a lot of non-synonymous point mutations. So just some caveats here. This is a big cross-sectional study, so these are broad correlations where we are supporting some theory and experimental predictions in the real world. So I think this is valuable, but of course, we don't know individual time series. These are not controlled experiments. This is sort of a snapshot of what's happening out in real patients. So that is a caveat, and we're hoping to do more actual patient time series to solidify these results. It does suggest the phage bacteria ratio as a diagnostic biomarker for disease severity. And again, this is cross-sectional. So what we'd really like to do is be able to, have a patient at admission and then predict, how likely are they to have severe or mild disease within 24 hours, so within the disease course, and can they then be treated more appropriately with antibiotics or not? And then finally, phage therapy is as promising as it was 100 years ago, but coevolution is something to keep in mind, and this is something that can actually happen in individual patients. And so I think this has implications for how we might design phage-based therapeutics. So that is the end of my part of the more detailed inpatient evolution. And I can pause for some questions and or hand off to Sayeed for a lot more applied diagnostics.
Yeah, I was going to suggest we take a couple of questions
Yeah,
now. If
yeah.
anybody has any questions, the chat is kind of quiet, so I hope people have some questions. I have one real quick one, if you don't mind. Oh, here comes something in the chat. But my really quick one is, what's happening in the environment outside of the patients? I mean, presumably these are host specific, right? They only infect the Vibrio,
This is
the
right.
phage.
So as far as we know,
So
yeah, sorry, I'll let you finish your
yeah,
question.
well, I guess we would only expect to find those phage wherever the Vibrio are in the environment. Are these same dynamics happening in the environment then, or do we expect something totally different given the concentration of the Vibrio or concentration of the phage or just environmental conditions?
Yeah, no, it's a great question. And right, so you're correct that I should have explained this, ICP1, this, as far as we know, this is a very specific phage for Vibrio cholera, right? So it's not resident in the microbiome. It's going to come in an infectious dose with Vibrio cholera. So it is in the environment with Vibrio cholera as well. So people will be, co-ingest both. And there were some formative studies in the early 2000s from another group in Bangladesh looking at phage concentrations in ponds around Dhaka, Bangladesh, and showing that it's actually anti-correlated with waves of cholera infection in people. So that in the environment, phages are perhaps suppressing Vibrio cholera in the environment such that, your people are less likely to get infected when there's a lot of phage in the environment. Now, those studies were a long time ago, and I think there's potentially some issues, so it's worth revisiting. And unfortunately, there's just, I think, in the cholera field, there aren't a lot of people working in patients and simultaneously working in the environment to actually make those connections. So these studies are often happening in isolation. So I think a challenge is to actually bring that together, because the environment certainly is going to be important as well.
Okay, interesting. There was one question in the chat. Maybe we'll just take this one real quick and then move on to you, Saeed. Vance, did you want to ask your question, or shall we just read it?
Uh, well, here I'm looking at it too. So,
The question is, what is the sensitivity of your antibiotic test? Is it possible that antibiotics outside the detection range could have had an influence on the phage
yeah,
to BC ratio?
yeah, this is
And then for
actually,
you.
maybe Saeed can answer this. I mean, this was in Eric's group that these assays were done. So there certainly is a limit of detection, limit of quantification. We are only looking at 3 antibiotics that we could quantify reliably. So there could be unknown exposures to different antibiotics that we weren't measuring. So certainly this is, there could be other factors that were not measured. I think that's fair. And then does VC have CRISPR immunity? Generally not. And what's interesting is there's actually a phage that does have a CRISPR system that targets one of the Vibrio cholera defenses. So are starting to analyze that now to see if there's sort of these layers of defense and anti-defense and so on. So we're looking at that now, but I don't have results for you yet.
Okay, thanks for the question in the chat. I'm going to suggest we move on and we can open things up for other questions as well. Saeed, go ahead.
All
Do
right,
you
so
have a comment? Okay.
I'm gonna just keep sharing for simplicity. In the meantime, Saeed, I don't know if you wanted to comment on the antibiotic quantification.
Yeah, I think like we mentioned, like we did the SMS. It's pretty sensitive, uh, but we don't look at the sensitivity of that that way. Like look at the percentage of sensitivity.
All
Okay,
right,
so
the the next slide deck?
Yeah.
Okay. All right. So I think you can take it away.
Yeah.
Can I go to the next slide?
Yeah.
So in the next few slides, I'm going to present our cholera diagnostic projects, where we are aiming to address the challenges imposed by the virulent phages on cholera rapid diagnostic test. So this slide is basically showing how the early detection of cholera outbreaks can reduce the mortality and morbidity. So let's look at how cholera outbreaks occurs. So initially, it starts with the introduction of Vibrio cholerae, the causative agents of cholera, to individual host, and within the host, it gets amplified, and then host shares the cholerae in the environment. And this phase leads to localized transmission, where few people in a community are infected, and this localized transmission leads to epidemic or pandemic, where large group in a communities are infected, and this leads to loss of mortality and morbidity. However, if you can detect the outbreak at the early stage, especially in the localized transmission stage, you can take preventive measures, and then you can prevent this epidemic or pandemic, and it can ultimately reduce the mortality and morbidity. Can I go to the next slide? So let's look at the different methods of cholera diagnosis. So microbiological culture is the gold standard for cholera diagnosis, but it takes at least 24 to 48 hours to get the results. And you also need skilled staffs to do this, and also need well-equipped laboratories to perform these microbiological cultures. PCR is also considered as gold standard, is quicker than culture. However, it is expensive, and it needs well-equipped laboratories and a skilled person to perform it. So you know that cholera outbreaks frequently occurs in resource-limited areas, so there is a lack of well-equipped laboratories and skilled persons to do that. So there is a high demand of rapid diagnostic tests for detecting the outbreaks. However, there are few rapid diagnostic tests available right now for detecting the cholera. However, the performance varied largely. So it has been shown recently in a review paper that the sensitivity varied from 31 to 100%, and the specificity from 60 to 100%. So the biological reasons for this variation are largely unknown. So our lab kind of work with one of the biological reasons that might affect the performance of the RDTs. So in 2018, our lab conducted clinical studies in Bangladesh, and in this study we showed can I go to the next part?
Oh, yeah.
Showed among diarrheal samples positive by nanoliter qPCR or Vibrio cholerae, the odd that a rapid diagnostic test was positive was reduced by 89% when there was a lytic bacteriophage present in the stool samples. So based on this finding, can I go to the next slide? So based on this finding, we hypothesized that when a cholera patient is co-infected with Vibrio cholerae and virulent phage, as an enhanced RDT that detects bibliophage as a proxy for Vibrio cholerae will increase the sensitivity of the RDT without compromising the specificity. So let's look at the hypothesis pictorially. So in the current RDT, there are several parts you can see here. You have sample pad that helps to take the samples from the sample tube. Then you have conjugate pad, where you have detection antibody conjugated with the gold particle. And then you have nitrocellulose membrane, where you have test lines specific to your target organisms, in this case, Vibrio cholerae. And you have a control line that specify whether your test is valid or not. And at the top, you can see the absorbent pad is kind of facilitated the capillary movements of the samples throughout these strips. So if you have a samples with Vibrio cholerae with no phage, what happens, the Vibrio cholerae from the samples is moved through this and is bind with the detection antibody that's tagged with gold particle. And then this complex will move through the steps due to the capillary reaction, and then if it is specific to that organism, it will be captured by the antibody that printed in the test lines, and you can see a line in the test line. So if you see a line, the test line is a positive for vibrio cholerae, and if you don't see, it's negative. However, if you have a sample where you have vibrio cholerae and the phage, you frequently get false negative results like that. Maybe due to like the, like as Jesse mentioned earlier, there's a predator-prey relationship. Then phage eats the bacteria, the lower the concentration of the bacteria, and you get the false negative results. So in our enhanced RDT, can you go to the next one? So in our proposed RDT, we hypothesize, we propose that if we can include one more test line in the current existing RDT, that is, the test line is specific to the virulent phage SP1. So this phage, can you go to the next? So in the samples where you have the vibrio cholerae and no phage, this RDT will detect vibrio cholerae as well, like the previous or existing RDT. However, in the scenario, can I go to the next? The scenario where in samples you have vibrio cholerae and the phage, where you get the frequent false negative results with the current RDT, you can detect this phage as a proxy for vibrio cholerae. So to address this hypothesis, we develop monoclonal antibodies to targeting the different structural proteins of ICP1 phage, and then we assemble those monoclonal antibodies in different combinations on RDT prototypes and optimize the assembly. And this figures is showing like the optimized RDT prototypes one. So basically, we found like the sandwich format of WRF122 monoclonal antibody and WRF monoclonal antibody 2 monoclonal antibody. What that means, WRF is the major head proteins of the ICP1. So WRF 122 monoclonal antibodies. So what we did, we printed that monoclonal antibody in the test line in this prototype, and we also used the same monoclonal antibody as a detection antibody. We conjugated that with the core particle, and I put that at the conjugate pad. And then we tested this prototype with ICP1 prep, and we had the ICP1 prep, and we boiled it in different time points. So this figure is basically showing the performance of this RDT prototype in different times after boiling. So you can see like when we don't boil the samples, these prototypes cannot detect the phage in the test lines. However, if you increase the boiling time, you can see the test line get intense, and it gets maximum to like 10 minutes or 10 minutes. Can I go to the next slide? So then we look at like we prepared like ICP1 prep, and we did the different dilution of ICP1 and look at like what's the limit of detection for this RDT prototype after 10-minute boiling. So we found like the limit of detection for this RDT prototype was 1/10⁷ PFU per ml is biologically relevant because in the median ICP1 prep is 1/10⁸ PFU per ml we found. Can I go to the next slides? So our aim to detect the phage ICP1 from the stool samples. So we had 3 stool samples, dilated stool samples. These are cholera negative, ICP1 negative. We spiked the ICP1 in this stool prep, and then we look at the performance. You can see like after 10 minutes boiling, for every samples that's positive, but from this figure, you can see there is an inhibitory effect of the stools for all 3 stools. So we try to like increase the boiling time, and like you can see like after 20 minutes and 30-minute boiling, you can see that kind of like overcome some of the inhibitory effect of the stools. So for the stools, we kind of went to like 20-minute boiling instead of 10-minute boiling. And then we also did the electron microscopy to understand what happened after boiling. So in these figures, there are 6 panels. Basically, A, C, and E is showing the panels. Those are the electron microscopy images So A is for no boiling samples, C is 10-minute boil samples, and E is the boil samples after 20 minutes. B, D, F, the same things that represent is the zoom in version of A, C, E. So if you look at the B panel, there's a no boil sample. You can see after labeling with the ORF 122 monoclonal antibody, few spots there. However, after 10 minutes boiling, you can see in the D panel, there are lots of spots, and similar things you can observe at the F panel, lots of spots there. So it's indicating that somehow when you don't boil the samples, the epitopes we are targeting with that monoclonal antibody is buried maybe with some other proteins, or it could be inside of the capsid. So that get exposed when you boil that, and it's boiling just increasing the abundance of the epitopes that we target with the monoclonal antibody. So in the next steps, we combine this optimized assembled prototypes with the normal existing RDTs. So we included the second test line, that monoclonal antibody specific to SP1, and we started a clinical diagnostic study at ICDW, set up that, and in this study we aim to include 500 ideal patients, hoping that 20% of the samples are cholera positive, and we aim to evaluate the performance of this enhanced RDT prototype against the existing RDT that is Crystal BC, called KIT, and using and as well as with the gold standard reference PCR and microbiological culture. So we recently finished that clinical studies, and we are currently analyzing the data, and we expect to increase of at least 20%, 15% sensitivity of the enhanced RDTs with this when you compare with the gold standards or other RDT existing RDTs. So these samples from this clinical study will be useful, especially this is the initial optimization of this prototype. So we have all the samples. If based on the results we get from this clinical study, we can optimize further these prototypes. It's basically some of the technical optimization, such as increasing the concentration of the monoclonal antibody in the test line, increasing in the conjugate pad. So this is important. So this is the overall update for the diagnostics project that we're conducting right now.
Hey, fantastic. Thank you, Saeed. Was all of this mentioned in the paper itself, or is this separate work?
It's separate paper.
Okay.
We just published that recently.
I
Sorry.
didn't remember that in the paper. I wasn't sure if it was in there.
Sorry, we should have
Oh, that's
shared it.
okay.
So the paper got
It's fine.
accepted
I think it's a
recently.
fascinating dimension of this. That's really fascinating how the phage can undercut the rapid diagnostic test. And we have at least one person in the audience who works on evolution of resistance to diagnostic tests. We're almost out of time, so I just want to see if anybody has any questions. In general, there are a couple in the chat, but I'd love to hear from somebody in the audience if anybody has a question they'd like to ask. And if not, we can go to a question or two in the chat.
We have a question here.
Okay,
Hi.
great. Thanks, Jeff.
Aha.
Familiar face. I'll just be a blur. Hi, I'm Julie. My question is on the feasibility of boiling to be folded into clinical protocols, especially for rapid tests. It's usually at point of care. What does boiling look like in that context?
Yeah, that's a great question. We try to do that because it's a rapid diagnostic test, and it's going to be used in the remote areas. So we you want something as simple as it is. So our original aim was to develop that way. So test that samples without boiling everything. Unfortunately, it didn't work. So then we thought let's boil the sample and see whether if we did not do the epitope mapping in this study, so that's the lacking of the study. So we didn't know where the epitopes is. So based on the ELISA or like based on blot, we went to like these RDTs. So interestingly, like I went to Bangladesh to work on developing this one. I worked for like 1.5 months, and then I was about to come to year back. And then just after 1 week before, and then I just thought like, let's boil the samples and see what's happened. Because so fortunately, the figure you saw the other one, I saw like after boiling is the test line intensity is increasing. So that's how we are now, but like there is a room to develop that one. I guess like in the next step for optimizing that one to find out another epitope that is like at the outer surface of the phage head, and then there's also abundance, so that we don't need to like go for boiling one. So this is the first step I would say like for developing this these RDTs.
Cool, thank you.
Okay, one last question, if it's a quick question from anybody. And thank you guys for just speaking up. Just speak up if you have the question. Otherwise, we'll just take one from the chat. There was a question from Ermi. Did you also test a phage cocktail or maybe more than one phage? I think maybe there's some confusion about the context here, that it's not a treatment that you're investigating. It's an investigation of naturally occurring phage.
Exactly.
Yeah, but I can answer that. So maybe you wanted to ask like whether we tested with other phage, because Jesse
Yes.
mentioned about there are other lytic phages that's specific to biblioquality. Yeah, so I didn't show that because of like time limitation, but we tested with the negative controls. We used like ICP2, ICP3, and biblioquality itself as a negative control, and this prototype did not like cross-react with that one, even if it boiled for like 10 minutes, 20 minutes. So yeah, so we tested that negative controls.
Okay, fantastic.
Yeah.
Okay, we're going to need to wrap up because we're at the top of the hour. I pasted in again the information on our Evolutionary Medicine Summer Institute and the topic, which is highly relevant to what we were hearing about today: overcoming resistance, harnessing evolution to control cancer, infectious diseases, and pests. Please check out that link before this chat disappears, and we hope to see some applications from folks in the audience. And with that, I want to thank Jesse and Sayeed again for a really great talk, and congratulations on the great paper. And the great collaboration you guys obviously have across different labs to see this through. So congratulations. Thank you.
Awesome. Thank you so much. Thanks for the invitation, and thanks everyone for being here.
Great. Okay. See you guys at the next one. Thank you.